Bismillahirrahmanirrahim. My name is Laiba Nasir and the program is BS Zoology. My topic name is DNA sequencing. So let's come to the point. I will discuss further more topics which include definitions, overview, methods of DNA sequencing, diagnostics, forensics. So come to the first point. DNA sequencing is the process of determining the sequence of nucleotides within a DNA molecule. Every organism's DNA consists of a unique sequence of nucleotides. Determining the sequence can help scientists compare DNA between the organisms, which can help show how the organisms are related. Then overview. This means that by sequencing a stretch of DNA, it will be possible to know the order in which the four nucleotides bases adenine, guanine, cytosine and thymine occur within that nucleic acid molecule. The necessity of DNA sequencing was first made obvious by Francis Crick's theory that the sequence of nucleotides within a DNA molecule it relates directly influence the amino acid sequences of the proteins. Sequencing methods. There are two main types of DNA sequencing. The older classical chain termination method is also called the Sanger method. Newer methods that can process a large number of DNA molecules quickly are collectively called high throughput sequencing (HTS) techniques or next generation sequencing (NGS) methods. Sanger sequencing. The Sanger method relies on a primer that binds to a denatured DNA molecule and it initiates the synthesis of a single-stranded polynucleotide in the presence of DNA polymerase enzyme. It using the denatured DNA as a template. In most circumstances, the enzyme catalyzes the addition of a nucleotide. A covalent bond therefore forms between the 3' carbon atom of the deoxyribose sugar molecule in one nucleotide and the 5' carbon atom of the next. This image shows how this bond is formed. First, guanine and this condensation reaction and next phosphodiester bond. This image shows the difference between labeled primers labeled DNTPS and di-terminated NTPS. Basically, this image shows a schematic representation of a di-terminated sequencing. There is a single reaction mixture carrying all the elements needed for DNA elongation. The completed reaction is run on a capillary gel. The results are obtained through an analysis of the emission spectra from each DNA band on the gel. A software program that analyzes the spectra and presents the sequence of the DNA molecule. Uses of DNA sequencing. Traditional chain termination technology and HTS methods are used for different applications today. First of all, Sanger sequencing is now used mostly for de novo initial sequencing of a DNA molecule to obtain the primary sequence DNA for an organism or a gene. The relatively short rates coming off an HTS reactions 30 to 400 base pairs compared to the nearly 1,000 base pair rates from Sanger sequencing methods make it difficult to create the entire genome of an organism from HTS methods alone. Occasionally, Sanger sequencing is also needed to validate the results of the HTS. Then come to the point, diagnostics. Genome sequencing is particularly useful for identifying the causes of rare genetic disorders. For example, while more than 7,800 diseases are associated with a Mendelian inheritance pattern, less than 4,000 of those diseases have been definitively linked to a specific gene or mutation. In one particular case, sequencing the genome of a child suffering from a severe form of inflammatory bowel disease connected to the illness to a mutation in a gene associated with the inflammation, XIAP, while the patient initially showed multiple symptoms suggestive of an immune deficiency, a bone marrow transplant was recommended based on the results of DNA sequencing. The child subsequently recovered from the ailment. This image shows the whole paragraph of the DNA sequencing. This image shows this paragraph is totally related with the DNA sequencing. I think you understand the important points of DNA sequencing which can be discussed in above. Thank you so much.